Hi everyone. I'm really happy to introduce Christoph Metzner. He's currently a postdoc at the Technical University of Berlin, and he's also a visiting lecturer at the University of Hertfordshire, where he did his postdoc previously with Volker. Christoph has been developing a model using NetPyNE for a couple of years, and I think he's about to publish the first paper using NetPyNE where we are not co-authors, so we're very happy about that. And yes, he's been really active in computational neuroscience in many different fronts. So I'm really grateful that he accepted to give this talk today. I'm really looking forward to it. So thank you so much, Christoph, and welcome.
Yeah, thanks very much for the nice introduction, Salvador, and thanks for the opportunity. Well, hi everyone. A bit unfortunate that I don't see everyone as usual, but I think we're all kind of used to it now. Yeah, so today I want to talk about this project where we looked at the effect of schizophrenia-associated gene variants on gamma band oscillations. And well, I want to start with, well, most people end with this slide. I want to start with it to acknowledge everyone that's contributed. And well, foremost, there's Tuomo Mäki-Marttinen, who now started his own group at Tampere University in Finland. And he's really the one that developed that framework that we use to put these schizophrenia-associated gene variants into biophysically detailed models. And then Gilly Carney and Hannah McMahon-Cole, two very talented undergrad students who did an internship with me and helped implementing the model. And then Volker, who's my former postdoc advisor and with whom I still collaborate a lot. And so a bit of introduction. So over the last years or decades, drug development for psychiatric illnesses hasn't really progressed very much. Well, a part of that problem is that there's a very large gap between the clinical symptom-based phenotypes on the one hand and the neurobiological and genetic vulnerabilities. So we don't really know how this wealth of knowledge that we have now on neurobiological and genetic vulnerabilities translates into symptoms. And therefore, in the psychiatry, a lot of people have started research that centers on the identification and validation of biomarkers. And the hope there is that these biomarkers, well, relate more closly to these underlying neurobiological changes, so that we can get this mapping more easily with these biomarkers. And these biomarkers could be, I don't know, behavioral things or neuroimaging with fMRI or measurements with EEG or MEG, for example. And then there's, well, there were also a relatively new movement in psychiatry where people use more and more computational modeling. Well, because they've realized that, well, computational modeling is ideally suited to provide this link between this genetic, molecular, cellular level and the biomarker or the symptom level. And in a recent review paper together with Tuomo, we basically argued for, well, we don't not only need like computational psychiatry, we need more biophysical psychiatry where we have really biophysically detailed models where we can integrate these low-level changes to make sense of what's going wrong at the whole brain level. And yeah, and today I want to basically present this framework that Tuomo's developed when he was a postdoc in Gauta Enewald's lab, where we use data from genome-wide association studies and put them into biophysically detailed models of biomarkers or endophenotypes. So the question is, how can we integrate this data on genetic vulnerabilities into models of biomarkers? And what we propose there is that we take the results from these genome-wide association studies, and we can then look at these risk genes. And with these risk genes, we can hypothesize on the functional role of genetic variants, but we can also use them to build models of, for example, ion channels or ion transporters, and put these into neuron and synapse models. And then we can use these models to model the effects on neuron function or the effects on circuit function, ultimately on behavior, that these risk genes or these genetic variants have, and constrain that by animal experiments or studies in humans. And so together with Tuomo, we did that, and basically the starting point was a biophysically detailed single cell model of a layer 5 pyramidal cell. And you see this beautiful image from the open source brain of that cell here. So it's a cell model developed by Itai Hay in 2011. It's a very detailed model, so it has roughly 200 compartments, Hodgkin-Huxley type dynamics, and has 10 ion channels or 2 sodium channels, 5 different types of potassium channels, 2 different calcium channels, and an H channel as well, and also models the intracellular dynamics. And it captures a wide range of dendritic and perisynaptic active properties. So it has these, I don't know if you're familiar, so layer 5 pyramidal cells have these 2 different integration zones. So they have these somatic integration zones where they get this input from basically sensory input on your sensory cortex, but also have this integration zone in the apical dendrite where they receive more top-down contextual information. And this model basically captures these integrations of these 2 types of inputs in the model. And we use that model then to incorporate these genetic variants. And the way we did that is, well, first we looked at genome-wide association studies. So, for example, this one by Ripke et al. From 2014, and looked for schizophrenia risk genes. But well, adding on to that, we also looked at genes that show a significantly different expression in schizophrenia patients. We used data from the Norway biobank, data on blood, so on gene expression in blood samples. And we focused on genes encoding ion channels and ion transporters. So a couple of genes, these are genes coding for calcium transporters, calcium channels, sodium channels, that's a small conductance potassium channel, and the H channel. And so these are all genes that encode for channels or transporters that are included in this layer 5 pyramidal cell model, and they are likely to be expressed in layer 5 pyramidal cells. And then we went on and went to the functional genomics literature and looked at the effect that these types of variants have on the ion channel kinetics. So then we can basically use this information to put the effect of the variants into our biophysical model. And the way we do that is these genome-wide association studies variants, they change the offset and the slope. So in Hodgkin-Huxley type model, you typically have activation and inactivation functions. So these are usually sigmoids or something like that. And these variants then change the offset, so they can shift. Let's say that the blue one is the normal channel, and the variant would then offset it either to the left or to the right. But it can also change the slope of the sigmoid. And that's we then basically for all these genes and many different variants, so I think in total it's more than 100 variants. In what I'm going to present later, I used 86 of these variants. All these come with a change of the slope and the offset of a certain ion channel. And for the expression data, we basically interpret the differences in expression as changes in the maximum conductance of the ion channels, and included it that way. So one caveat for this, especially for the inclusion of the genome-wide association study data, is that, well, I said we looked at data from functional genomics, but actually the data for these SNP-like variants you find in with genome-wide association studies, well, that data doesn't really exist. So no one has looked at what a SNP-like variant does for these genes. So instead, what we used is information from in vitro studies of more extreme genetic variations of these genes. So that also includes loss of function mutations. But then, because these are more extreme variations, we had to apply a downscaling procedure. So for example, a loss of function mutation sometimes could result in, if you just put that into the model, could result in the cell not firing any action potentials at all. And we assumed that this would be unrealistic for these genome-wide association study type variants. Yeah? And so we applied a downscaling procedure. And basically the central assumption here is that the effects of these SNP-like variants can be represented as scaled down versions of those of the more extreme variants. And furthermore, that the emergence of the full psychiatric disease phenotype is basically a result from the combined effect of a large number of these subtle SNP effects. Yeah, so every little variant only has a very small subtle effect, but if you combine them in the right way, or the right ones, then you get a disease phenotype. And then first, we looked at, well, if these variants change the excitability of single cells. So we then saw that, well, yes, some of these variants can alter the calcium response to a short stimuli to the soma. So as you can see here, there's a brief 5 milliseconds input current to the soma that results in the cell firing one spike. And here for different variants, you can see basically the calcium response. So it's the calcium and the change of calcium on the y-axis. And you can see for some of these variants, for example, these three affect these two affect high voltage activated calcium channel, and this one affects a low voltage activated calcium channels. And this is a calcium transporter gene. So they affect the calcium response in relatively strongly, while these two, that's for a sodium channel and this for a potassium channel, don't change the calcium response that much. And what you will always see here is, so the blue line is always the control case, and the other colors are different scalings of one variant, where you scale them, basically multiply the effect with a factor. And we did that in this case in both directions. And then you can see, for example, here the blue line is in the middle, and the ones that have a positive factor go in one direction, and the one negative one have basically the opposite effect. But you can see some of these variants change the excitability. And you can also see that the steady state firing is influenced. So here you have an FI curve. And again, some variants affect the FI curve relatively strongly, and others like this one don't affect the FI curve. And last but not least, in these single cells, these variants also produce differences in the inhibition of a second apical stimulus. So you have this paradigm where you have two short inputs to the apical dendrite, and typically when you present the first one, that means that the second one is, depending on the stimulus interval, the second one gets inhibited. So the second one doesn't produce a response, or you need a relatively strong stimulus to cause that response. And you can see that some of these, so this threshold that you need for the response is here on the left, on the y-axis, sorry, and depending on the stimulus interval here. And you can see that some of these variants increase or decrease these thresholds. And you can think of this paradigm as a single cell version of it's called a pre-pulse inhibition, and that's a biomarker for schizophrenia. So the pre-pulse inhibition of startle reflexes, where you have puffs to the eyes of mice or rats, and they two in a row, and the first one is a very low intensity puff, which basically goes unnoticed, but it inhibits the startle reflex to the second one. And you can think of that as like a single cell version of this paradigm. That's well studied in schizophrenia. So then we went on and put those layer 5 cells into a small network model and looked at delta oscillations, because changes to delta oscillations have also been described in schizophrenia. And we see first we as an input, we give noise that is modulated by sinusoidally modulated by a certain frequency and a low frequency. And basically we then look at the power of the frequency component that corresponds to the input modulation frequency. And you can see that these variants can change this. Well, first of all, you see a resonance. Basically, the strongest power is in the delta band, so between 1 and let's say 3 or 4 hertz. And you can see that these variants can modulate this power. And interestingly, if you combine some of them, you get quite strong changes to the power here. And then if you drive the model purely with noise and look at the power spectrum, then you can also see, so that's a semi-logarithmic plot here with the frequencies here and the power on the y-axis. And if you then zoom into this low frequency band, you can see that, again, the blue is the control, and the other ones are scalings of, that's the combination of variants we saw before. And you can see that they can relatively strongly increase the delta power here. Basically, overall, we see that these variants and in the right combinations can alter these biomarkers, or can alter properties that are considered biomarkers for schizophrenia. But then we wanted to put that into larger network models, and as I said, the full Hay model has 200 compartments and 10 ion channels and many differential equations. So it's computationally too expensive for large network simulations.
Christoph, can I stop you there? Sorry.
Yeah, sure.
Like most models are based on mice, right, or rodents.
Yeah.
And their layer 5 is at least, like, even physically twice the size of non-human primates or primates. So what implications do you think that has on what you were talking about?
So sorry, I'm not sure.
So basically, if you, and I can send you a figure after the talk, like rodents, subcortical, layer 4 layers, infragranular layers, are twice the size of non-human primates and primates. And it's the other way around for primates. Like, so do you think, like, what implications does
I mean,
that have?
so I mean, it might have implications for the processing layer 5 cell does, right? But I don't think it has implications for how we incorporate the genetic variants into this cell.
Yeah,
right?
no, I don't think
yeah,
so.
yeah.
But what does, so this model is based on rodents, right?
Yeah, the Hay model is a I think it's rat data. Yeah.
So given that the size difference is so large, and I'm not like attacking you or anything, but would it be worth to try something in non-human primates?
Yeah, I think so. Well, actually, what I wanted to look into, well, hopefully soon is so. I've started to look into this layer 5 pyramidal cell and this well, integration of apical inputs and somatic inputs in the cell more. I had an intern working on that, but there have been, I think, 2 or 3 papers now from Francis Skinner's lab where they have models, biophysically detailed models in the same order of compartment and so on from human layer 5 pyramidal cells. And they do find some differences, especially I think related to the H currents and so on. And I think, well, going towards using this model in that framework would be very, very interesting.
Okay, yeah, I'm just very interested because, like, I know layer 5 projects to the thalamic, like, modulatory regions, like pulvinar matrix, the sensory matrix, and all that.
Yeah.
And layer 6 projects back to the sensory parts.
So I mean,
so yeah,
coming back to you, so I think the single cell properties are a bit different. So it would be interesting to look at that only, like just focusing on the single cells. But then also
like even like Christoph, I really enjoyed your talk, by the way. But in rodents, you don't even have inhibitory neurons in the medial geniculate body, which is the main relay for the auditory system. And in non-human primates, it's like 30% or something like that. I forget, but so I think we have a need to develop parallel models of, like, this is all based on rodent data. And I think we do need to develop, primate models. And I'm not chastising you or anything, but
yeah. I mean, I agree. Yes, and I think especially this study from Francis Skinner's lab where they compare this Hay model with their model based on data from human layer 5 cells. That's really interesting. Yeah.
I actually don't know that. Would you mind sending it to me?
Yeah, I can.
On my screen, it just says you, but do you see my name or?
Yeah, I can. Yeah.
Okay.
I can send you. Send it to you later. Yeah. Okay. So
thank you, Cristoph, and great talk, by the way.
Okay.
Thank you.
Yeah. So basically, we wanted to put that model then into larger networks, and therefore we had to reduce it. So what we did is we built a reduced 4-compartment model. You can see here on the right. So that's again the full Hay model, and the 4-compartment version has one compartment for the soma, 1 compartment representing the basal dendrites, and 1 compartment represents the apical dendrite until it starts to branch out, and the 4th compartment is basically where it branches out into the tufts.
Sorry, Cristoph, to stop you again.
Yeah.
Do you agree with the notion that all pyramidal neurons in cortex have their apical dendrites up in layer 1?
Uh,
Or no? Like, you know the literature much better than me. That's why I'm asking.
So I'm not sure if I know the literature better than you, to be honest. So I don't know.
Because
I
this
think
is
most of
perfect.
them.
This is I totally agree with this figure. But do you think then what's been proposed lately is that layer 1 is the main controller of all cortical operations, or no?
Maybe, I mean, yeah. I mean, so you're referring to what is one paper from Matthew Larkham's group, or?
Yeah, he's the main proponent of it, but there is other ones, and I talk to my anatomist friends who do, monkey anatomy, and they do say that basically all dendrites reach to layer 1, no matter what layer are you in, like layer 6, 5, of course 3 too. So do you think that inhibitory matrix is controlling all cortical operations? Okay, you know what? I leave this for the discussion. So yeah.
Okay. yeah. Okay, great. Yeah, let's do it. Let's do it in here.
Sorry, just to that point, I think this paper we used, Peter Budinger 2018, they show like several cell types in layer 5 and layer 6 that only reach layer 2-3, not layer 1. I can send you the figure for that.
You mean in the CSD? In our data?
No, in a paper that we used to build the model, Budinger
Okay.
had a circuit diagram and several of them. I can send you later.
Okay.
Okay, so yeah, so we reduced this model to 4 compartments, and still has all the ion channel, and it fairly well reproduces the properties of the full model. So passive properties, input currents just to the soma, number of spikes it produces, input currents to the apical dendrite, produces 1 spike, combination of inputs to soma and apical dendrite, and also the FI curve fairly well. And so now, basically with this model, that's the one I then used to build this model to look into these gamma oscillations in schizophrenia. So now we're giving a short detour about gamma oscillations in schizophrenia. So I mean, you probably all know that gamma oscillations are found throughout the brain, and you see them in all the sensory areas. When you get sensory input, you see it in motor areas, hippocampus, and other areas. And there are some hypotheses that they underlie local communication processes. So there's this communications of coherence theory by Pascal Fries. And they have been linked to things like perceptual grouping, attention-dependent stimulus selection, working memory, and a lot of other things. But in schizophrenia, you see that gamma oscillations are significantly impaired. So in task contexts, so in, for example, in Gestalt perception, you see a reduction of gamma oscillations, but also at rest. And at rest, you can even see that there's an effect of the progression of the disorder. So, for example, here, this is gamma power at rest in chronic schizophrenia patients, and you see reductions in a lot of regions. But for example, first episode psychosis patients show a mixture of regions where the power is reduced and regions where power is increased. And in at-risk subjects, you see that they mainly show increased resting state gamma power. So, but overall, gamma oscillations are impaired. And if you look more specifically in the auditory system and at auditory steady state response tasks, you see reductions in Evoke power and intertrial phase coherence. And for those of you who are not familiar with these auditory steady state response tasks, usually what you do is you represent click trains. So each of these lines here represents one click of, let's say, 1 kilohertz. And a 40 hertz click train just means that you have a presentation frequency of these clicks of 40 hertz, so 25 milliseconds between each click. And then you can see, if you look at the LFP, or in humans at the EEG or MEG, that these auditory areas, the network basically follows this 40 hertz input with a 40 hertz oscillation. So here's the filtered version and a nice peak of 40 hertz in the power spectrum. And in schizophrenia, you see for these auditory steady state responses that they're, the most robust finding is a reduction in the gamma band. So here you see that's 40 hertz input. And if you then look at the power for the control group, there's a clear, strong peak at 40 hertz. But in the schizophrenia group, there's a much lower peak, so there's a reduction in gamma power here. And there's also some other differences. So, for example, there's an increase in the beta band. So, yeah, we see also auditory steady-state response differences in the beta band and also in the delta band. But I want to focus on the gamma band here, because that's the most robust finding here. And so we wanted to look at now at the effect these genetic variants have on the gamma oscillations in these auditory steady-state responses. Yes, Peter, you.
Sorry again. That's so cool, because we find the same, that beta, alpha band steady-state responses or phase research or even amplitude is increased in schizophrenia, as opposed to the other bands, which likely indicates that, you know, they are more internally focused than externally focused, if Pascal Fries is right. Like, you know, he advocates that alpha beta is top down and
yeah. That's
anyway,
yeah.
Sorry.
Okay, so then we built this very simple EI networks. We have a population of pyramidal cells and a population of basket cells, so fast spiking evolving positive neurons. And obviously for the pyramidal cells, we use this reduced version of this layer 5 model that we developed. And then we use from the literature, we use a model of a cortical basket cell. And we have sparse and random connectivity. So the glutamatergic connections have AMPA and NMDA receptors, and the GABAergic ones, GABA-A receptors. And both E and I cells get this ASSR drive. So basically, every 25 milliseconds, they get a bout of input, and they also get some background noise. And then we can simulate an LFP, and you see the LFP for this 40 hertz click train here, and then we can make a Fourier transform and look at the Evoke power here. And of course, that's the model that's implemented in NetPyNE, Salvador talked about before. And we put the models into this reduced version as I described before. And first we do some network tuning, so some control conditions. Well, first we calibrate the background noise so that we have spontaneous firing that's comparable to A1. And then we simulate this auditory steady-state responses for a number of different frequencies, so for 40 hertz, 30 hertz, and 20 hertz, because these are the ones that you most typically use in human EEG MEG studies. And we see that, well, comparable to experiments, that the power for this 40 hertz auditory steady-state response is highest, and then it goes down to 30 and 20. And also interestingly, in healthy human subjects, you have this relatively strong 40 hertz component, even when you drive with 20 hertz. and that matches some experimental and also previous modeling studies. And then as a further control, we also reproduce some data from optogenetic manipulations of fast spiking interneurons. So the study from Cardin et al., where they optogenetically drive basket cells with an input of a certain frequency, so light pulses at, let's say, 25 milliseconds or 33 milliseconds and so on, for different presentation frequencies, and also find that in our model, driving these basket cells in the gamma range, so between, let's say, 30 and 50, 60 hertz, increases power more strongly than when we drive them with other frequencies. So now we're kind of well, a bit confident that the model really, the control model is a good model of what's happening during auditory steady state responses in humans. And now we look at the effect these variants have on the power in the gamma range. So, well, again, we see that these variants can produce gamma band power deficits. So what you see here is blue is the control network. So the reason why you're seeing a box plot here is we not only do one simulation per variant. What we do is we simulate 20 different subjects by changing the random seeds for the connectivity. So we get different connectivities. And then on top of that, for each of these subjects, we simulate 10 different trials by changing the random seed for the background noise. And then as you do in experiments, we average those 10 different trials for each subject and end up with basically a response for one subject. And here in these box plots, you see the box plots for the 20 different subjects. And this blue line is the power, the gamma power of the control network. And then here in orange are all the variants. So in total, it's 86 variants, and in orange you see the variants that affect the high voltage activated calcium channel. In green, the ones affecting the low voltage activating calcium channels. The red ones affect the H current, and the purple ones the transient sodium channel. And well, you can see, well, the single variants change the gamma power, but they typically, their effect is relatively subtle. And you can also see that, for example, these variants of the SNC1A, which affects the sodium channel, they don't seem to have an effect at all. But there are also some variants that relatively strongly reduce the gamma power, like this one or these ones. So if we look at those in more detail, so you can see they relatively strongly reduce the gamma power. But so now we want to look at combinations of these variants, because as we saw before, these combinations sometimes their effect was much stronger. So we do 3 different combinations. So first we take the 2 variants here and this one here that produce the strongest reduction. And we add a third one. Well, that's not the third strongest, but for this combination, we didn't want to have 2 variants that belong to the same gene. But in the last version, we combine them. And when you combine them, our assumption is that their effect, because they affect the same gene, that they superpose linearly. And then we look at these 3 different combinations. So here again, so here you have the 3 combinations, power for them. And that's again this single variant that produced the strongest effect. So you can see that combinations produce stronger effects. And while they analyzed that a bit further, and they kind of superpose linearly. So if you have one variant that reduces it, the power, let's say, by 20, and one that reduces it by 10, the combination reduces power by roughly 30. So it's a linear Yes, Peter.
Sorry, Christoph, to stop you again, but
no worries.
So in our hands, when you look at monkey data, usually power differences are not that great between, you know, states or whatever. But what we figured out is that stimulus-related phase similarities, which is like some people call ITC, some people call other things, is super different between different states. So is there a way to model that?
So yeah, I don't have it on my slides here, but I also looked at intertrial phase coherence. Yeah. And well, these variants don't change the phase coherence that much.
They don't.
No.
That's super. Now, Christoph, we have to look into that because in monkey data, they are so, like ITC is so different and power is not different. So, we can share a data set with you and like maybe we could talk about it.
Yeah. I mean, so the things in data, human data with EEG, you find both. You find robust changes in gamma power and in phase coherence. Yeah.
But Christoph, that's because any phase reset, like let's say you reset auditory cortex, that will cause an amplitude increase. So there is no real difference because synchrony in the human brain, because of volume conduction, causes amplitude increase. But in our data, that's really local. You can really see a difference.
Okay.
And let me know if you want to look at it.
Yeah, that's interesting. Let's just wait for the next slide, and then we can talk about
okay. Sorry to interrupt again.
But that's fine because then so typically when people model these changes in these auditory steady state responses, they look at changes at synapses. So so we compared the effect of the variants to schizophrenia-associated synaptic alterations. So one is an increase in the basically the time course of the inhibited postsynaptic currents. So that is in schizophrenia, you have a reduced expression of this GAT1, which is an enzyme that basically removes GABA from the postsynaptic receptor. And if they are the expression of this GAT1 is decreased, then basically GABA stays longer at the receptor and the time course is longer. So it takes longer until the inhibition wears off. And we looked at that. And another finding in schizophrenia is reduced GABA levels. So we have a reduced expression of this GAT67, which is a precursor of GABA. So there's less GABA available at the synapses. So you have a reduced strength of the inhibition. So we included those two alterations into our model, and then we compared our the effect of the variance to these two changes. So here you see again that's the single variant with the strongest effect. Here is this combination of these four strong single variants. And here's a reduction of GABA levels, and here's a reduction of an increase in the iPSC decay times. And you can see, well, they are kind of comparable. It also, in these types of model, it's a bit hard to really, well, constrain how strongly you have to reduce, for example, the GABA levels, what's realistic there. That's a bit hard to do, but they have a similar effect. And now with respect to these to the phase coherence, so as I said, for our variants, we don't find an effect for the phase coherences, and I don't have the figure here, but it's in the in the preprint. But for the reduction of GABA levels, and especially for the changes in these iPSC decay times, you find a relatively strong reduction of the phase coherence. So
that's very nice. Christoph, can I stop you there?
Yeah.
Again, like, I'm sorry, like, I'm being a meanie, but why do you think it's GABA associated? Like, what's the evidence for that based on your data? And the reason I'm asking, and I'm not just, again, not being mean, I am, but schizophrenia has been tied to GABA deficits, you know, all sorts of
yeah, dopamine, NMDA.
yeah, dopamine, GABA, yeah, like, so, like, does this provide evidence that it's GABA associated, or as opposed to other things?
I mean, so basically what we show here is that if you just look at the gamma power, then it could also be the genetic variants. If you look at the phase coherences, then it looks more like it's one of those GABA effects, but something we haven't incorporated in this model, but what we could do is NMDA receptor hyperfunction that has also been linked to gamma oscillations. There's some paper from Bart Ehrmantraut's group where they show that, well, if you model dopamine changes to dopamine in schizophrenia, that also affects the gamma power.
That's what I was.
Coherence.
So dopamine affects gamma. I didn't know that actually. So that makes sense, because that's the leading theory.
So I think the reasoning is something that dopamine changes some properties or modulates the potassium channels, or some types of potassium channels that then are important for the gamma oscillations.
Okay.
But
that makes sense. No, that would be cool. Yeah.
So the thing is, overall, so I've looked at these especially these gamma auditory steady state responses now for the last 5 or 6 years in different models, and
I actually did it in cats, Cristoph.
Okay. So for me, it's I think if you just look at the reduction of gamma power and maybe a change in the phase coherence at gamma alone, that doesn't constrain the models well enough to really pinpoint, is it more GABA, is it more dopamine, is it more NMDA, or maybe so you have to look at other parts.
Cristoph, this is very important. So the model cannot predict the mechanism, or can it? Or
you just with gamma power, you can't constrain the model enough to pinpoint the mechanism, because there are so many.
Okay, Cristoph, this is super important for grants and for future research. What would you need us researchers to?
Yeah, that's a good point. That's something I've been thinking about as well. And so here I'm at the conclusion, so I can go back to the.
I'm gonna shut up now.
No, that's it. I mean, it's very interesting. So to this slide, so there are some things. So basically, everything I presented in the later part was just looking at the power here compared to here, right? But you can see there's also differences here to here. However, that's only this one study, and there's 2 more that show that, whereas the other 12 or 15 gamma auditory studies response studies don't show this increase in 20 hertz for the schizophrenia group. But you can also see 40 hertz power at 20 hertz drive, so at beta drive, is also higher in the controls compared to the patients. And 20 hertz power here is higher than here. So it seems to be a reduction of gamma and an increase in beta in both of these conditions. And well, basically trying to have models that for example, can replicate all those 4 things might constrain it more to pinpoint the mechanism. Or you might even want to think of how can I construct a stimulus that's not only a 40 hertz or 20 hertz, but maybe a combination, a superposition of some different oscillations that, with this one mechanism, it produces this kind of power spectrum, but with the other mechanism, it should produce something else. And that's something I wanted to start to look into that. So how can I construct a stimulus that can tease the mechanisms apart? So that I can see the difference in the power spectrum in the end. Yeah. So I had a grant proposal where I proposed something going in that direction, but that didn't get funded because I think the main reasoning was, well, why do you need to do modeling? You can all do that with optogenetics, so just do it with optogenetics.
You can do optogenetics in humans, so like what's the rationale? I
obviously disagree with that rationale, but and especially if you want to tease apart, I don't know, maybe 4 or 5 different possible mechanisms or combinations of them, the number of experiments you'd have to do is really high, and you could very easily do that in the computer model. But
Cristoph, if you want, I can share my latest grant with you on schizophrenia, but it's monkey, but like, maybe you'll get ideas or whatever.
That would be really interesting. Yeah, that would be great. Thank you.
So, and did I send it to you, Salva? The schizophrenia one? No?
Not yet, no.
So maybe you could share Christoph's email address with me, and I could share it with both of you, and then like you'll see.
Yeah.
Because I think there is a lot in there. I mean, it's horribly written because it's written by me, but I think there is good ideas in there.
That sounds really interesting. Yeah. Great. Okay, so to just briefly conclude then. So I presented this framework to incorporate these data from genome-wide association studies and also studies on gene expression in schizophrenia into biophysically detailed neuron models or network models. And we saw that these variants can change single-cell excitability, integration of somatic and dendritic inputs, delta oscillation power, delta resonance, and then in the last part in this model that they can decrease gamma band power, and especially that these combinations of variants can produce relatively strong reductions that are comparable to previously considered synaptic alterations. And with that, I'd like to thank you for your attention. And if you have questions,
thank you.
Ask them now, or you can also send me.
Are you physically in Berlin?
I'm physically in Berlin, yeah.
How is the weather?
Terrible.
Oh, okay.
Yeah.
Same as in Hungary, I guess. I'm from Hungary originally, so. Okay, cool. Cristoph, thank you. And Salvador will share your email, and then I'll send you the grant.
Yes, great. Yeah.
This was a great presentation. Thank you very much.
Thank you.
Thank you so much, Cristoph. And if you have any questions from the students or anyone else.
I have a question. I'm joking. I think I had enough.
Yeah. May I ask a random question?
Yeah.
About these gamma increases and decreases at different stages of the disease. Is it about positive and negative symptoms, or no one knows? Because usually at the late stages, negative symptoms predominate. Is it like low gamma negative?
That's true. That's true. So positive symptoms have been linked to increases in resting state. But it might also be so. The thing with the patients with chronic schizophrenia is that you cannot be sure that it's really well, the progression of the disease or medication effects, because chronic patients are well, almost all of the time they're stably medicated, and could be just an effect of the antipsychotic medication. But yes, there have been some links of changes in so these differences were in resting state gamma power. So and I think increased resting state gamma power has been linked to positive symptoms, and that basically also ties in nicely with that you have these increases in the first episode psychosis patients, but you only have decreases in the chronic patients.
Yeah, thank you. Does it match what happens with gamma rhythm on Nmda antagonists like ketamine? Because in ketamine anesthesia, as far as I understand, gamma is largely increased. It dominates everything. And what happens in sub-anesthetic dose, I don't know, in fact.
Yeah, it's similar. So yeah, so sub-anesthetic ketamine increases the resting gamma power. So that matches with the.
Okay, thank you.
Any other questions? Okay, well, thank you so much again, Cristoph, for your fantastic talk.
Yeah, thank you. Thank you again for your interest. Thank you.
Thank you.